This video provides an overview of the methyl red Vogues Proskauer test. This test starts with a basic tea soy broth with glucose present, and we're looking to see what type of end products are produced, whether they are an acid end product due to mixed acid production, or whether it's a neutral to maybe even slightly basic end product. We're basically looking for a pH change through various reagents. This is a medium that's split into two tubes. So you put the same bacterium into both tubes, the same species of bacterium, incubate, and then you add the reagents. So here on the left we have methyl red reagent that's been added. And the test result of it staying a bright red indicates that it is a positive for the production of mixed acid, so the pH is dropped. Now we did the exact same incubation here with the same organism. We just simply added a different reagent at the end of the incubation period. I'm going to zoom in here just a little bit. And here we added the VP reagent. The VP reagent is a combination, Vogues Proskauer reagent, a combination of two reagents, and it's somewhere around 8 drops of A to 4 drops of B, but that can vary, so always check with your instructor on that. And that needs an incubation time of somewhere from 5 to 10 minutes, maybe even up to 15, and that varies a little bit depending on, again, just the type of particular type of VP A and B reagent that we use. Now, you should never see a positive for both tests. So what I see here on the left is a positive for methyl red, and that tells me this organism makes acid end products. And I got a negative for the VP test, which also confirms that the end products are not neutral to basic, because I didn't get a positive here. I got a red color in methyl red, which indicates the end product is acidic. Now, taking a different bacterium over here on the right, I now have the same setup. I have a methyl red test against a VP test. I took, again, a different species of bacteria, but put the same species in each tube. It's, again, a broth that's split in half with glucose. And I added methyl red in one tube to test for acid products in that particular tube. And then I added the VP A and B reagent to test for a pH change, again, neutral to maybe even slightly alkaline. The product that I'm looking for here, actually what I'm looking for in particular is 2,3-butanediol. However, the particular product that this test picks up is a precursor to that acetone. So I'm testing for acetone, but if acetone is present, that's going to indicate that instead of mixed acids produced at the end of this fermentation, you're going to see a 2,3-butanediol that's produced, or it could be also acetyl methylcarbinol. Again, it's key that the VP reagents are added in the correct amounts in the right order. And I compared a couple different methyl red negatives here, just to note on these methyl reds that it clearly needs to be a bright red color. So I'm going to move this other one over here, pull this out. This is actually a negative methyl red. It's more of an orangey color. It needs to be bright red to be considered positive over here. Here you can see a clearly, this is from the test I started with, a clear bright red color, which is going to indicate an acidic end product. Anything other than that, so little to no red, or here's almost kind of an orangey red, is going to be considered a negative. And I should again see negative. This is a different species of bacteria, which is a slightly different change, but again should see a negative MR test going along with a positive VP test, and the VP test appears positive by the formation of a red ring at the top of the tube after 15 minutes. So this gives, again, an overview of the MR-VP test, which covers the type of end products that are produced at the end of glucose fermentation. You're either looking for acidic end products or neutral end products. And I say neutral, it can be slightly basic. And you're doing that by adding methyl red to one tube to pick up any acidic products that might be there, and then in the other tube with the same bacterium, adding the VP reagents to pick up a rise in pH. And that is the overview of the MR-VP test.